All right, welcome everyone to our ISACB webinar. And I'm Hannah Cebull. I'm part of the Young Investigators Committee, and I'll be the co-chair for today's session. I am a postdoctoral fellow at Emory University School of Medicine, and my fellow co-chair, Fabien Kawecki, is a postdoctoral fellow at the University of Bordeaux.
So thank you, Anna, for this introduction. It is a pleasure to co-chair this webinar with you. So I have the great pleasure and honor of introducing to you Professor Elaine Roche. She is an internationally recognized researcher at the Department of Mechanical Engineering and the Institute for Medical Engineering and Science at MIT. She received her PhD at Harvard University under the guidance of Professor David Mooney and Professor Connor Walsh. And before joining MIT, she conducted research as a postdoctoral fellow at the National University of Ireland, Galway. And now as a PI, she explored the intersection of mechanical and biological therapy delivery. And our group uses tools including computational modeling and image-based biomimicry to study the interaction and mechanical effects of implantable devices on the cardiac tissue. So today she will present her fantastic work on novel devices to repair or augment cardiac function. So on behalf of the Young Investigator Committee of the ISACB, I would like to thank you again, Elaine, for accepting our invitation. And now the screen is yours.
Thank you, Fabien, and thank you, Hannah. It's a pleasure to be here this morning. My time. I guess not for everybody, but yeah, thank you for inviting me to present the work here. It's a pleasure. All right, well, I'm going to jump straight into it. Thank you for the kind introduction, Fabien. So I'm an associate professor here at MIT. I started my group in 2017. And this is kind of the overall design process that we follow in our group. We develop implantable devices, mainly for cardiovascular, for augmenting cardiovascular function, and we have some devices recently to augment respiratory function. This is kind of the linear design process that we tend to follow. Design of device, we simulate and test them, and then ideally we translate the devices to the clinic. We get input from industrial and clinical partners, and if successfully translated, we can have our technologies adopted by industry and by the clinic. But where we spend a lot of time is in this loop here where we simulate and test, and we do a lot of in vitro, in silico, ex vivo, and in vivo modeling, and I'll talk through some of those today. And really, when we spend a lot of time iterating through this simulate and testing loop, we find we can establish some insights into the biomechanics of the disease and some of the mechanical influencers of the disease. We really look at how we can augment the mechanical function and how we can recapitulate the mechanical function of systems on the bench and in animal models. Broadly, my research can be divided into 3 strands. The first is mechanical augmentation of organ or system function. We have some tools that can augment cardiac function and also recently augment diaphragmatic function. I won't spend too long in that area today. I'm going to focus on strands 2 and 3. Strand 2 is more local, structural or biological repair of tissue. Again, I'll focus on the heart today, where we use devices to locally deliver therapy to the heart. And then the third research strand is multimodal simulations of biomechanics. Like I mentioned, these can be benchtop or computational models, or indeed we can use devices to recreate disease models in vivo in animal models. And I'll spend a bit of time today talking about that strand as well. For each of the clinical vignettes, I'll try to step through why we do it, what we do, who the team that works on it, how we do it, what is the impact of it, and what are the future steps. When I say clinical, sorry, I mean preclinical vignettes. Okay, so I mentioned I won't spend too long on the first research strand today. Some of it is previous work that I did, and others is focusing more on the respiratory system. So I'm going to focus on the second and third aim. Starting with structural or biological repair of tissue, I'm going to start by describing a device that we developed to deliver therapy to the epicardial surface with multiple dosing and to simultaneously provide cardiac support. This is based on work that I started during my PhD with David Mooney, and we continued it in the lab here at MIT. One of the problems that we address here is myocardial infarction, and specifically missed MIs that progress to heart failure. And the number is quite high at 20% within 5 years for these missed MIs. There is a lack of therapy to prevent this progression to heart failure after a heart attack, and you can see how the ventricle dilates and there's remodeling. For therapies that are under evaluation, one-time delivery is only possible, and there's difficulty retaining therapy on the heart and delivering multiple doses. This is a team of PhD students and postdocs and an MD that have worked on the project I'll talk about now. So we specifically looked at delivering FSTL1, a protein or bioagent that has been shown in the past in previous work to encourage regeneration of the heart if delivered to the epicardial surface after a heart attack. Previously, multiple doses delivered locally hasn't been possible. And we use this kind of therapeutic reservoir that can be attached to the epicardial surface of the heart. It contains a gelatin sponge, and there's a refill line that goes to a subcutaneous self-sealing port. So we can refill over time and deliver multiple doses of FSTL1. And we studied this in a rat model of MI, where we did a permanent ligation of the left anterior descending artery. We have 5 groups in this study, and we had one group with no treatment, so just a myocardial infarction, a sham device without any biological agent, 1 dose of FSTL1 delivered via the gelatin sponge, 2 doses, one that was refilled through the device, and then 3 doses, 2 refills through the device. Interestingly, what we found is that more dosing tended to lead to better function over time. This shows the fractional shortening over time, and you can see that with the myocardial infarction only, you get low 20% fractional shortening. Even with the sham device, you get a significant increase, but then you get a trend towards not a significant difference in the shortening with multiple doses, but a trend towards improved function. So we can really use this platform to study the timing of dosing, the timing of delivering therapy post-infarct, and the number of doses. You can see histologically there's quite a difference between the control group, the sham device group, and then the different numbers of doses here, where you can see the ventricular wall is thicker and healthier as you give more doses. If you quantify the scar size as well, you can see a decrease with any amount of this bioagent delivered. So the scar size is smaller. So we're excited about using this platform to evaluate different types of therapies. We've made this platform a little bit more functional too. Sorry, there's questions coming in. Do you want me to answer them as I go, or should I wait until the end?
I think we'll wait until the end, if that's all right, unless you I guess it's whatever you prefer.
Yeah, of course. Yeah, I can answer this one as I go. I don't think I actually showed the timeline here. My apologies about that. But yeah, we delivered this therapy at the first refill at 1 week and the second refill at 2 weeks. And we brought the animals out to 6 weeks. Yeah, so we've made this a little bit more functional, so we can combine the delivery to the epicardial surface with triggered delivery, and we use soft robotics to achieve this. So we have a pneumatic chamber as well as a loaded tough hydrogel that's actually adherent to the tissue surface. So now when we pressurize the upper chamber, we can cause compression of this gel layer and cause release into the epicardium on demand. So we can control both the timing and the spatial delivery of this, of the whatever cargo that we're trying to deliver. We can also incorporate sensing into this, so we can sense things like arrhythmia and deliver antiarrhythmics on demand. And there's no sutures required for these types of reservoirs either. Here you can see that we see a significant difference depending on the degree of compression that we give to this reservoir, and the length of time for which you compress. So you can really tunably control the release of therapy from these reservoirs. We're also looking at using the mechanical restraint of epicardial patches. And here we're working with a collaboration with the Zhao group here at MIT. And we're doing direct ink writing of bioadhesive patches, which have these unique kind of strain stiffening properties. And you can see the printing process down at the bottom here. So you can very finely control the sinusoidal pattern, and you can orient the patch in such a way that you can anisotropically reinforce the mechanical properties of the heart. Shown here on a pig heart, you can see when you put on the patch, you can control the longitudinal strain while maintaining the circumferential expansion and not impeding filling. So this is kind of an interesting research direction, just looking at the mechanical restraint of the post-infarct heart. Okay, so I'm going to switch a little bit, and within the same research strand, I'm going to discuss another reservoir. This time we're using it not directly on the heart, but to deliver therapy for diabetes. We call this a soft transport augmenting reservoir. And this is a team, again, that worked on this project, a group of postdocs, MDs, and graduate students. And this work was done in collaboration with Professor Duffy and Dolan at the University of Galway in Ireland. The clinical problem here is type 1 diabetes when pancreatic cells don't make insulin and the blood glucose is uncontrolled. The treatment limitations are that insulin delivery can be challenging, and implantable devices are challenged by a foreign body response. So there's a lot of research towards making a bioartificial pancreas or a macro encapsulation device to deliver insulin producing cells. This is what a macro encapsulation device would look like. It could be islet cells or beta cells inside the capsule. It has to sense glucose and deliver insulin. So there's a design consideration with a selectively permeable membrane here. Not only that, there's a semi-permeable cell containment barrier, but really, as well as glucose, or as well as insulin, it has to allow the transport of glucagon and somatostatin, and it has to ensure that oxygen, nutrients, and glucose can get inside to the cell therapy, but keep immune cells out. So there's this very precise design of a semi-permeable cell containment barrier. In addition to this, there's a process that happens over time when you implant such a device. In the beginning, there's kind of this acute immune response, and where you get neutrophils infiltrating, then you get macrophages and fibroblasts differentiating to myofibroblasts. You start to get collagen deposition over time and eventually get a dense fibrous capsule. And this impedes the function of such a macro encapsulation device because you can no longer sense glucose or deliver insulin from your cell cargo. Previous work in our group, we used simply mechanical actuation, intermittent mechanical actuation over time to modify the fibrous capsule response to implantable devices. So in this first proof of concept study, we did 2 weeks, and we implanted a reservoir subcutaneously in a rat model. There was a control group that was static and an actuation group where every 12 hours we would cyclically actuate the device for 5 minutes at a frequency of 1 hertz. When we looked at the end of those 2 weeks, we could see there was a decrease in the fibrous capsule in the actuated group, with the small n number admittedly shown here, but this kind of encouraged us that potentially this intermittent actuation could modulate the immune response. So we built another device specifically in this case for delivering insulin. The end goal is to encapsulate cells that produce insulin. For this first proof of concept study, we delivered insulin directly. So the device consists of 3 layers: an outer layer, an actuation layer, and a porous layer. And there are 2 lines, an actuation line, a therapy line. They go to 2 reservoirs. The upper reservoir is the actuation reservoir, where we can use either air or saline to move the entire reservoir. And the lower reservoir is therapy reservoir, where in this case we deliver insulin. This is what it looks like when we deliver the drug or the therapy. Here we're just using a dye so you can see where the drug fills, and this is what the intermittent actuation looks like. So the delivery of the drug is through passive diffusion. The intermittent actuation is once every 12 hours for 5 minutes, as I mentioned before. And we looked at a mouse model in this case. What we found was we could map the foreign body response to the blood glucose response very closely. So over time, when you immediately implant, you have no capsule, then you develop an acute foreign body response and then a chronic foreign body response. And if you look at the blood glucose change over time, you can show how the blood glucose change kind of maps onto this foreign body response. At baseline, you get a very sudden drop in blood glucose when you deliver insulin through the device. At 2 weeks, it's a slower drop, and you get less of a drop in blood glucose overall. At 3 weeks, you get even less of a change in blood glucose in response to insulin, and it's slower again. So this capsule is impeding the response. If you quantify this in terms of max blood glucose drop, you can see kind of a linear decrease over time in the amount of blood glucose change. We can map the fibrous capsule thickness that we measure histologically to the blood glucose drop, and it's a very linear trend. And then we can model over time how much the thickness of the capsule slows down the response and the drug release. So you can see over time, as you get a thicker capsule, you get a slower response and less of a response overall. So in this study, we had 3 groups. The first group was a control group. There's no intermittent actuation. The intermittent actuation is noted as IA here. Then we started one group where we did intermittent actuation daily. At 3 weeks, we stopped half of that group. And then we continued for 8 weeks with, again, actuation every 12 hours. And here is the blood glucose change in response to an insulin delivery at 3 days, 3 weeks, and 8 weeks. And the colors correspond to the timeline above. So at 3 days, both the actuated and the control group are pretty similar. You start to see a little bit of a less of a response in the control group, but quite a similar overall drop in blood glucose and a similar time to get that drop. Now, if you look at 3 weeks, you can see that the control group has a slower response and changes the blood glucose less in the 2 hours post-insulin delivery. At 8 weeks, the control group is still dropping to the blood glucose by the same amount. Or sorry, the actuated group is still dropping the blood glucose the same amount as at 3 days, while both the group that was actuated for 3 weeks and then stopped, and the group that was actuated for 8 weeks has a blunted response, doesn't drop the blood glucose as much, or very little in fact, and is much slower. So essentially what we're doing by using this intermittent actuation is extending the therapeutic lifespan of the device. So at 8 weeks with this actuation, we can get a similar blood glucose response to insulin as we do at 3 days. And this is shown here. So the baseline at day 3 is not significantly different from the max blood glucose drop from the 8-week actuation group. Meanwhile, the group that was actuated for 3 weeks and stopped has a much lower response, and the control group has a very low response. If you look at the time to get to a 30% blood glucose drop in the control group, over time in the study, you can see how it gets higher and higher, and it takes longer to get to the same blood glucose drop. Similarly, when you stop actuating at 3 weeks, you get this longer response time. But for the group that's consistently actuated, the time to get the same blood glucose drop doesn't really change over the entire 8 weeks of the study. So this is exciting. We don't use any drugs here at all. We just use this low frequency and low pressure motion every 12 hours. So we see a lot of uses for this. And this is a proof of concept for delivering insulin. Why does this happen? I guess, first of all, we looked at early response, and we counted the number of neutrophils in the control and intermittently actuated group. And at day 5, we see a significantly less neutrophils in the group that's actuated. If we look at the amount of myofibroblasts at 2 weeks, we see less in the actuated group as well. And this is stained by alpha SMA. The long-term effect is a little bit different. So we think that the long-term effect is more related to the collagen orientation. So you can see the control and the intermittent actuation group, the collagen deposition looks a little different. And we quantified this at week 2. And especially at week 8, you see a difference in the coherency and the orientation of collagen. Also in the actuation group, you see clearing of any infiltration into the macro encapsulation device. So this is kind of a multifactorial phenomenon that we're still looking into a little bit more, but early on you see a change in the acute immune response. You see a difference in the thickness of the capsule to a certain point, and then you see a difference in the quality or the orientation of the fibrous capsule at later time. Overall, we can extend the therapeutic lifespan. We can somewhat modulate the immune response just with motion. And now we're working with collaborator Professor Millman at University of Washington to load with stem cell derived beta cells so that we can actuate and see if we can enhance the glucose sensing and extend the therapeutic lifespan of this cell therapy. Okay, so I'm going to jump into research strand 3, which is multimodal simulations of biomechanics. I'm going to start with the development of a high fidelity cardiovascular in vitro simulator. This is quite different to the last research strand where we're delivering therapy. Here we're really trying to make better benchtop models. So what we did here, the problem that we were trying to solve is that it's quite difficult to replicate both the biomechanics and the anatomy of the heart in a benchtop testing model. The need is that heart models are potentially not recapitulating biomechanics or not representing the anatomy at a high enough fidelity. And the question we asked was, can we use our soft robotic approaches to enable better benchtop simulators? This was previous work that I had done during my doctoral work where we used a soft robotic sleeve to augment heart function. This was an implantable device. And then we thought, can we use similar approaches but apply them on the bench so that we can reanimate explanted tissue and create a high fidelity benchtop model? This is a team again. Clara recently graduated from the group, is now at Abbott Vascular, and this was the subject of her PhD work. So first of all, we published this work about a biohybrid approach to replicating heart motion and function. So we took a chemically preserved whole porcine heart. So we had accurate anatomical details. We removed the muscle tissue very carefully by following the myocardial fiber orientation. So we had this endocardial scaffold and retained all the intracardiac properties. And then we used soft robotic cardiac muscle to replace the removed heart muscle. If we can then combine the soft robotic cardiac muscle with the preserved endocardial structures, we can reanimate the heart, and then we can have kind of a longer lasting benchtop model with accurate anatomical details and very tunable, controllable heart motion. Both the septal wall and the left and right free walls can move, and you can replicate both healthy and disease conditions, as I'll talk about. This is a CT scan of the biohybrid heart in a long axis and short axis view. The endocardial tissue is shown in the lighter, and the soft robotic myocardium and muscles is shown in the darker gray. This is what it looks like here in real life, and this is a collaborative work with Dr. Hyunwoo Yuk and Professor Zhuan Zhao, who really helped us to develop a bioadhesive to couple the explanted tissue to soft robotic muscle. To really make this clinically viable or preclinically useful as a test bed, we had to optimize the design of the soft robotic cardiac muscles to get the accurate biomechanics and hemodynamics, and then figure out a preservation method for the endocardial scaffold so that it would last longer. We started a computational modeling exercise to optimize the design of the synthetic myocardium. We used a finite element modeling. We used a thermal analogy to describe the actuator motion based on experimental data. And then we have a fluid model where we can look at the ejection volume with different designs and the pressure generation with the contraction of the synthetic myocardium. And then we can look at tuning a wide variety of parameters to achieve physiological ejection fractions and physiological pressure generations. Here are some of the design parameters that we looked at, but this computational tool allows us to really screen a broad number of parameters. We looked at the density of actuators, how many of these we would need, and each of these actuators is a small balloon-like structure that's embedded in a silicone matrix. We can design them in different ways to get different amounts of linear contraction and radial expansion. So how many of them would we put in the matrix? What type of actuators would we use? What material could we make them out of? Would we put a mechanical restraint or a pericardium around the matrix? And how exactly would we place the actuators to ensure the optimal motion and biomechanics? And with this tool, we could screen a number of these to guide the design process, which was a lot more efficient than building multiple of these on the bench. I won't go through all the details of this, but we discovered that the optimal design in terms of recreating physiological parameters was using two different types of soft actuators, flat and 3D McKibben soft artificial muscles. We had two layers, one circumferentially placed and one helically oriented, so you could recreate both the twisting motion and the compression motion. With this design, this was screened against multiple other designs, but with this design, you could get a left ventricular volume displacement of over 80%, and you could get a rotation, subapical rotation of about 8 degrees. So with both of these, we could quite accurately model, at least in silico, the motion of the heart. We also looked at coupling this to a fluid model, because if we want to advance this at some point to using it with blood, we can't have stasis or stagnation of blood in the chamber. And if we can take our mechanical model and couple it to a fluid model, you can look at the displacement of fluid. This is with a control design where we just have circumferential actuators, and at the bottom here is an optimized design. And what you can see here, sorry, I'm not sure if those videos played too well for you, but basically what we're looking at is velocity streamlines over one contraction cycle. The blue velocity is higher velocity and the red is slower. So you get some stagnation or slower velocity blood in the control, and you get less in the optimized design. And if you look at this over the different stages of one cardiac cycle, you can see that during the ejection phase, there's there's higher velocity in the optimized design. There's less areas where there's low velocity and less stasis. So that would lead to less clotting. And we can do further fluid modeling of this, where we can look at time average wall shear stress and endothelial cell activation potential to really quantify how much potential clotting you would get. And the optimized design has better has less thrombogenicity overall. So then we had to build it and make sure that our models made sense. So here you can see just a slow motion video of this design and its ejection fluid, just so you can visualize the amount of fluid it ejects with one cycle. But we could, with a contraction, get a left ventricular volume displacement of 70% and then a systolic pressure generation of 130 millimeters of mercury. We had to, again, see if this was useful for clinical testing. And we focused here on a left ventricular simulator. We took the biohybrid heart that I've described with soft robotic cardiac muscle and endocardial scaffold from a porcine heart. We connected it to a mock circulatory loop. And in this loop, we can vary left atrial and arterial compliance, and we can vary preload and afterload. We can also sensorize, so we can have a pressure sensor in the ventricle, the atrium, and we have a flow probe on the aorta and on the left atrial side. This is what it looks like in the lab. So you have an electropneumatic control system where you can control individual actuators in the hybrid heart, data acquisition system that acquires data from your flow meter, pressure sensors, and flow probes. And you can also have an endoscopic camera in the heart, because you can perfuse this with a clear fluid that still simulates the viscosity of blood, but then you can have direct visualization of things going on in the heart. You can also use echocardiography on this simulator. So here's the motion of the heart, and then here is kind of the pressure generation that you can get in the aorta, the blue line, left atrial pressure, and the left ventricular pressure. So you can kind of tune these to be close to physiological. We really wanted to look at whether we could use this and generate functional valve motion. So here you can see the aortic valve moving on echo and under direct visualization, and the mitral valve. Mitral valve can be hard to replicate in these type of benchtop simulators, but here you can see the motion of the mitral valve. And we wanted to see if we could recreate pathology with the mitral valve. So the acute mitral regurgitation we mimicked by rupturing the chordae. So by cutting the cords here, severing the cords. And what you can do is you can have moderate or severe mitral regurgitation. And you can see we can quantify the amount of mitral regurgitation here by looking at mitral flow and the amount of regurgitant flow specifically. So from baseline to moderate to severe, you can get different levels of mitral regurg depending on the extent of cord cutting. Then we wanted to see, can we look at repair of the mitral valve? And we looked at, in this case, a trifecta valve from St. Jude and implanting it surgically. Pre-treatment, you can see that the motion and the coaptation of the leaflets isn't that good. Post-treatment, you can see this nice motion and leaflet coaptation here. You can quantify this as well with the various sensors. So you can see at baseline, this is a mitral flow with severe mitral regurgitation, and then with the tissue valve in place. And you can basically quantify the regurgitant fraction with this repair methodology. We also quantified interventional tools. We looked at a MitraClip procedure. And we did a surgical repair, so we can really look at various mitral valve interventions. And I think the value of this simulator is that we have septal wall motion and ventricular wall motion, and we have direct visualization of how these look. So this work on the biorobotic heart has led to a lot, and Fabien is here. We have started a collaboration with Fabien and Dr. Leroux on right heart models and tetralogy of Fallot, which is really exciting. We've also looked at mitral valve biomechanics with Professor Wu's group at Stanford, and we've used this as a dynamic phantom for coronary imaging at MGH. We're moving towards using it as a total artificial heart. Clinically, we've looked at various valvular disease and interventions at Boston Children's Hospital, and we're also generating models of LAA closure in collaboration with Beth Israel Hospital. This is also being used in industry. We're working with Philips Research for developing an imaging phantom. We're working with Boston Scientific for developing dynamic LAA models as well. Okay, so I'm going to switch gears and talk about another type of soft robotic sleeve that enables hemodynamic and biomechanical mimicry of left ventricular pressure overload and aortic stenosis. The problem here is aortic stenosis, a disease where the aortic valve is narrowed and doesn't allow enough blood through. So you get these high pressure gradients, and it can lead to heart failure where the heart can't fill, so diastolic heart failure. And the models for testing interventions here again are limited. So we wanted to look at how we can accurately mimic aortic stenosis with high controllability and also mimic congenital heart defects in vivo. This work we wanted to do in an in vivo model, and we wanted to reduce the mortality with these types of models. There's a big team working on this, and we work with Professor Chris Nguyen, who's now at the Cleveland Clinic. We formed this sleeve that goes around the aorta. We used thermal forming methods to make 3 pockets that could be selectively actuated. And we put them in this fabric band that could be wrapped around the aorta. And each of these pockets could be pressurized to various degrees. The fabric constrains the expansion, and you can modify the amount of, you can modify how tightly the band is wrapped around the aorta. These are filled with saline to mimic aortic stenosis. We did a study in vivo in a porcine model where we put this sleeve around the aorta. We measured cardiac function and implanted the device at day 0. And then day 6, we used MRI to measure cardiac function and aortic geometry. And at day 8, we did a catheterization where we could measure the pressure volume loops. And we did echo to monitor cardiac function and flow. What you can see here is the pressure volume relationships over time. So this is baseline, the blue loop here, and then you can see when you actuate one pocket, you mimic bicommissural valve disease, and you get a higher pressure generation but less volume displacement, and that becomes more severe as you selectively actuate more pockets. So with 2 active pockets, you get a higher narrow PV loop. And if you actuate all 3 pockets, you really get a high level of stenosis. And this you can tune in vivo over time. The end goal for this is that we create a chronic pressure overload model, and we can induce cardiac or left ventricular remodeling and potentially develop a model of HFpEF or pressure or due to pressure overload. You can measure very accurately the flow with MRI at different levels in the aorta with different amounts or with different numbers of pockets actuated here. So you can look at tricuspid, bicuspid, and unicuspid aortic valve disease. Next, we're looking at taking clinical data from patients with aortic stenosis. And then we're printing both the left ventricle and the aorta of these patients with a soft 3D printing method. This is all in vitro. Then we make a patient-specific left ventricular sleeve and a patient-specific aortic sleeve. And then on the bench, we can tune both the aortic pressure, but we can also tune the compliance of the ventricle. And then we can test different treatments on the bench for patient-specific models. So if we look at CT of a number of patients on the top here, some of them with bicuspid aortic valves, we can recapitulate the effective orifice area with our in vitro models that are 3D printed with our sleeve around them. We can use a die-score coefficient to quantify how close we are to the same area, effective orifice area. And then we can do multi-material printing to actually print the calcifications based on CT scans and really recapitulate the types of depositions these patients have. If you quantify effective orifice area, you can see that we can very closely match the patient and the model. And even with the multi-material 3D printing here is shown for patient 9 to 11. With this sleeve, you can vary ventricular compliance. You can also create the motion in vitro, this contraction and relaxation. And by tuning the amount of relaxation you do, you can modulate the compliance of the LV. So you can mimic remodeling of the ventricle as well as the aortic stenosis. So it can go from healthy to moderate to severe remodeling. You can also look at intervention. So we used a TAVR here and looked at how much we can increase effective orifice area and decrease the pressure gradient by implanting the TAVR here, and just shown for 2 different patients. Ultimately, we want to kind of validate this with clinical data. So this is kind of the first in vivo model that uses that can customize aortic flow patterns to model aortic stenosis and congenital valvular defects. This work pioneers the development of high fidelity models of cardiovascular disease for hemodynamic mimicry using soft robotics, and we hope it paves the way towards patient-specific modeling. It's a tunable high fidelity in vivo model of aortic stenosis, and we hope that with the combination with the patient-specific in vitro model, we can identify which patients would benefit from clinical intervention. We can test and develop novel therapies. And ultimately, with a chronic model, we can look at ventricular plasticity and timing of intervention so that we can figure out at what point relieving pressure overload can help the ventricle to reverse remodel. Okay, the last thing I'm going to talk about, if we have time, I'm just conscious of the time here. I'll go quickly through this because this is really the last kind of simulator that I want to bring up here. This is for single ventricle physiology. This is a normal physiology and a single ventricle physiology that's been repaired by a Fontan surgery. So these patients are born with 1 functioning ventricle instead of 2. And they go through 3 surgeries. The terminal or the final surgery is called the Fontan procedure, and they end up with, instead of 2 parallel kind of pumping systems, 1 in series pump. So there's 1 ventricle pumping systemically, and the return from the to the pulmonary circulation from the venous side is passive through this Fontan shunt. This leads to a lot of comorbidities, high pressures in the liver and liver damage, lung damage, low oxygenation. You can get decrease in heart performance, developmental and neurocognitive delays. And we're working with a group in Children's Hospital to really understand how the flow in this Fontan shunt is linked to breathing mechanics. So the IVC flow during inspiration increases, and during expiration, you actually get a decrease and you get retrograde or backflow towards the abdominal organs. This is, again, just like a typical biventricular flow here in the IVC flow rate, and with the single ventricle physiology, you get synchronization, with breathing, not with the cardiac cycle, and you get a retrograde flow up to 30 to 50%. And there isn't a ton of characterization of this Fontan flow pattern done to date, so we kind of wanted to tackle that and the mechanical influencers of this flow. This is a huge team. Again, it's in collaboration with Boston Children's Hospital and with Dr. Pedro Del Nido and a wider team at Children's. What we did was we recreated a benchtop simulator of the Fontan flow, including the respiratory mechanics, and a lump barometer computational simulator of this interplay between the systems. This is the physiology of the respiratory system, and this is our respiratory simulator. We have a diaphragm that's actuated by soft robotic muscles. We can generate abdominal and thoracic pressures, and we can inflate and deflate lungs. This is shown for visualization purposes. Now, if we overlay a flow loop into this in vitro simulator, we can put in Fontan a specific circulation, we can get the characteristic vascular anatomy of a Fontan patient, and we can get the diaphragm and chest wall anatomy from a CT scan. And then we can put a flow loop through this entire system, and the flow is now subjected to the thoracic and abdominal pressures. We can then simulate the vascular beds of the lungs and of the abdomen, and we can perfuse the fluid, or perfuse with the fluid with the viscosity and density matching blood. Now you can see as you move the diaphragm, you get this change in compliance, and you get this change in the flow of the system. You can visualize it here with movement of air in the system. So there's no pumping fluid through, it's all moving just because of the pressure changes, the transdiaphragmatic pressure change and the pressure change in the thorax. And what we can do then with our model is we can recapitulate inferior vena cava flow. So the flow in this shunt, we can mimic exactly how it is clinically. This is a patient IVC flow in blue, and this is our in vitro IVC flow. We can also mimic the pressures during inspiration, expiration, both in the thorax, abdomen, and also in the hepatic vein and in this Fontan shunt. So we wanted to look at the effect of different types of breathing. We can do it in this in vitro simulator, but we wanted to validate it clinically. So with a group of children's, we conducted a study on Fontan, people with a Fontan physiology, and we had them in the MRI breathe in different ways: normal breathing, shallow, heavy, slow, and fast breathing. And they use this kind of meditation app to regulate their breathing and follow certain patterns. We're very grateful to these patients. We scanned 20 of them, and then we can quantify both aortic and IVC flow, and we can quantify the amount of retrograde flow in response to different breathing patterns. We can look at chest wall motion to quantify breathing effort and respiratory rate. What we found was that the heavier breathing in these patients led to more retrograde flow. Similarly, faster breathing led to more retrograde flow, and this has adverse consequences, the more retrograde flow. And this really closely matched what we found in vitro in our benchtop model. So it was nice to validate this with a 20-patient study. We have multiple ongoing studies looking at the effect of exercise on this flow and looking at how we might modulate this flow with a extravascular support or with an implanted valve. But what we did show is that both breathing effort and breathing rate are independent contributors to backward flow in the single ventricle physiology. We looked at the impact of blood flow, blood pressure, vascular compliance, and breathing on Fontan flow patterns, both in vitro and in vivo. And then we collected a library of patient flow profiles, which we can now use to evaluate device performance in a heterogeneous patient population. We also can look at body position and the effect of gravity with our in vitro simulator, which is hard to do in vivo. We can now make recommendations of beneficial breathing for single ventricle physiology as shallow, slow, using the chest to breathe, and a supine position. And we're excited to continue work on this, and especially looking at the abdominal flow patterns with the group at Boston Children's. Okay, so I'm going to finish there. I probably am a little bit longer, but I still have time for questions. I hope that this process was kind of clear that like, you know, ultimately we want to translate to the clinic, but this time that we spend simulating and testing with these different modalities, in vitro, in silico, ex vivo, and in vivo, really can help us to establish insights and give feedback to the clinic and to industry, and how we can test these in higher fidelity environments, how we can test these devices. So thank you for your time. This is my wonderful team who do all this work that I presented here today. I mentioned them throughout, and I thank you to the funders as well. All right, thank you.
Thank you, Elaine, for this amazing presentation. So it's time now to open the discussion session for the next 20 minutes, I will say. So I encourage the audience to use the little icon to raise their hand, and then I will give them the microphone to ask questions, or they can also directly ask questions within the chat.
So I see one question in the chat about the durability or the lifespan of the biorobotic heart. Yeah, it's a great question. I didn't discuss how we preserve the heart tissue, but we did come up with different ways to increase the longevity. So we first we can fix with formaldehyde, but then decellularize to mimic. So we mimic the mechanical properties of the heart, and then we find these things can last for a long time. We have probably over a year is the one we've kept the longest, but we could still actuate it over that period of time.
Are there any more questions? I will have one.
Yeah.
So you within your presentation, you presented a 3D printed pericardial patch that you implanted on the animal heart.
Yeah.
I want to know, how did you stabilize the patch? Did you implant it on a curarized heart and then restarted the heart, or it was directly implanted within a beating heart? Because I imagine it's really difficult.
Yeah. So you basically want to implant we wanted to implant it during diastole. So we did it on a beating heart. You can temporarily kind of occlude to implant the patch, or you can rapid pace if you wanted to temporarily, but this adhesive works pretty instantly. So, you know, you kind of get it ready and then implant it in the diastolic phase.
Okay.
So, yeah, we did it on the beating heart. When do you think, or at what stage do you think clinical applications may start? Yeah, that's a great question. I think a lot of what I talked to today was further away from the clinic and more kind of like in the research stage. We have some other devices that are closer to the clinic, closure devices for septal defects, and some occluder devices as well, which are kind of closer to clinical adoption. These ones that I spoke about today, I mean, I think that the test beds, we're working with industry at the moment to deploy these simulators and test beds in industry. They won't be like clinically adopted, but they'll be used hopefully in industry to test therapies and technology. So they could be adopted by industry sooner, and the regulatory pathway and the path to commercialization is lower for them. But if the FDA, want to have high fidelity test beds, the hope is that these can provide that data. There's a question.
Yes, Craig has a question.
Ellen, great talk, really impressive work.
Thank you.
With the trainees on the call, and myself included, you've gotten funding from NIH, NSF, foundations, and industry. Can you maybe talk a little bit about your strategy when you talk to these different federal agencies, different foundations, different industry, about how you get your work funded?
Yeah, absolutely. I'm still learning. I think I set up the lab 5 years ago, but I think it depends on the agency. For NIH funding and for early career researchers, there's some kind of high risk, high reward funding mechanisms. And I didn't talk about the project where I have kind of one of those R21 Trailblazer awards, but for my diaphragmatic augmentation project, an implantable ventilator, I have funding for that project, which was kind of really like high risk, high reward idea.
Through NIBIB, is that right?
Yes, through NIBIB, yeah.
Okay.
My NSF funding is through EBMS. I have a career award through NSF, and for that it's on the biohybrid simulators. And there I think it's very important for the career award to have a research program that's integrated well with the teaching. So I use these simulators. The respiratory simulator I use in my class on respiratory pathophysiology. And then I teach a class on medical device design, and we use the cardiac simulators to test some of the technologies. So I think their, you know, integration of research and teaching is very important. And for that, I focus more on the technology, like how do we recreate the biomechanics of the heart and the diaphragm. I didn't talk about the diaphragm work today, but that's kind of for the career award. I have an EFRI award from NSF with Professor Zhao on catheter delivery and control, like kind of robotic control of catheters. Again, more on the technology side. So I think being like strategic about which agency to go to with various projects is helpful. The foundation award, so I have for the single ventricle physiology, I have funding from the Charles Hood Foundation for Excellence in Child Health Research, because this is for congenital heart disease. And then there's a group called Additional Ventures that specifically funds single ventricle research. So I have funding for them. So I think for disease-specific research, sometimes foundations are a really great place to get funding for those projects. So yeah, I mean, applying broadly, I definitely have not got all the funding I've applied for, but you know, applying broadly and trying to match the project with the best potential funder. Just being persistent, I would say, is key.
Great, thank you.
Of course. Yeah, I guess my work is very much at the intersection of engineering and kind of, you know, the more medical applications of it. So in some ways that's an advantage because I can apply to different foundations, but it has to be very tailored.
Are there any more questions from the audience or on the chat? Can see. I will have maybe one last more personal question, because for
Of course.
some people here know that I've got the chance of being part of your lab for a few months, and it was a fantastic experience.
No,
and for people who don't know Elaine, she's really great PI, and she's successfully managing a team of approximately 20, maybe more students and postdoc, which is amazing. She's so accessible. She takes time for all of them. It's so incredible to see that within science. And also in parallel of being a PI, she's a mother of 3 wonderful girls. And as a father of twins, I know that raising kids requires a considerable investment. So my question is, how do you succeed to combine your personal and professional life while keeping a competitive research level in ultra highly competitive institutions such as MIT? Please tell us your secret.
Oh gosh, I wish I could. I take it one day at a time. There's some days I think work wins, and sometimes family just needs attention. So I think I have good support structures in place here. You know, Fabien, you saw like the daycare is across from the office, so it makes it easy to visit the kids if they need it, bring them to the doctor, take them out if they need to. I have a husband that helps out a lot, which I'm fortunate for. And I think I try to involve the girls as much as I can in the lab. And if they're out, they come in and they hang out. The students are always great, and we'll show them around and show them the science. So, you know, I think they like being part of the environment, and luckily my group are accommodating of that. I just do the best I can to make it work, but I have very good support as well.
Okay,
yeah.
Thank you. That makes sense.
Never a dull moment, as you know, Fabien.
Yeah. Thank you, Ellen.
Of course, there is one question about the soft pockets of the aortic sleeve and the TAVR device. Yeah, that's a good question. So the sleeve is on the outside of our in vitro model, and then we create you know, a pressure overload or aortic stenosis, and then the TAVR, it was a self-expanding device that we tested, will open up, you know, the aorta again. I guess probably the question's getting at how you know, how much does that simulate the reality where it's more like a calcified valve? And I think there, you can tune the sleeve compliance, but you could also use the multi-material 3D printed models with the calcification on the valves if you wanted to really look at how the leaflets deform. And we are in a process of having realistic calcification deposits on those leaflets, on actual tissue leaflets. So that's kind of an ongoing project. In the ones I showed, it was like a 3D printed material, but we're really looking at mimicking the calcification properties on tissue leaflets as well. So that might be better for quantifying what happens when you implant a TAVR. The sleeve gives you the overall global hemodynamics, but at the local level, maybe a model like I just described could be better.
Great. Well, thank you so much for sharing your exciting work with us today. And before we close, we just wanted to mention that we're in the process of planning some future webinars. So if you know of any other early career investigators, or if you yourself want to share your work, please reach out to us. I believe you could respond to the email that you got for the registration for this webinar. And yeah, thank you so much for attending.
Thank you so much for having me. It was a pleasure, and thanks for the great questions, and I appreciate it.
Thank you so much, Ellen.
Thank you.
Thank you.
Bye.